So this division of Ascomycota, the fungi, so the fungi present in these sectors are called as ascomycetes, commonly known as sac fungi. So they play an important economically important fungi or family, we might say. For example, your aspergillus. So aspergillus, it is mostly they are red, brown, or blue-green molds, and they cause food spoilage, and they also produce powdery mildews that attacks the plant leaf, and fungi that causes chest blight or Dutch elm disease. These are caused by ascomycetes. So this, when it comes to your structure of your ascomycota, they contain a fruit cell, then a stalk-like structure called as conidiophore, then your primary stegmata or philadia. It is a flake-shaped cells from which the center part is called as your vesicle, through which your primary stegmata is formed. At the tip of your primary stegmata, a secondary stegmata is formed, and along with this, a chain of conidia is attached to your secondary stegmata. Then comes your reproduction. So the reproduction, asexual, is by means first your suitable food cell or a vegetative cell is being found or it has been seen. They act as a base for your formation of your conidiophore. So then from there, the conidiophore gets developed. Conidiophore is developed from conidiophore is developed. There will be an enlargement at the apex region. They form a bulb-like structure, which is called as conidiophore. Upon this conidiophore, they form an enlarged structure. This conidiophore gets matured. This maturation will help for their spore development, inside which the spores gets developed. From there, your stegmata is produced, which is your primary stegmata. So the stegmata, as I said, they are flake-shaped cells. This is arise from your vesicle. The center part is vesicle, and these phyllides are spore-producing cells from which your secondary stegmata is formed. A stegmata is formed through which conidiospores gets attached to the tip. These conidias are non-motile, unicellular, and pigmented, mostly green, brown, or black. During favorable condition, these spores gets detached and goes to a suitable nutrient-rich or moisture-rich content region, and they gets germinated, like a germ tube is produced, and through which your mycelia gets produced, and sorry, not mycelia, your food cell is produced, and the cycle or food cell or vegetative cell is produced, through which the cycle repeats. This is your asexual reproduction. Then comes your sexual reproduction. So as same as your asexual, here a slight difference is seen. Here two strains are used. So that is your heterothallic hyphae structure. First, your same as your asexual reproduction, a vegetative cell is developed from which two heterothallic hyphaes are formed. These heterothallic hyphaes are of ascogonium and antheridium. Your male is your antheridium and your female is your ascogonium. So these two are formed at a favorable condition. These two gets fused, or there will be a contact between your male and female hyphae. This contact this contact is beginning with your plasmochemy, your cytoplasm fusion, from which through this fusion a sterile hyphae is produced, or a sexual structure is produced, which is your sterile hyphae. A fruiting body called as ascocarp is produced. From this ascocarp, inside the thick walled structure is called as ascogonium. Inside the ascogonium, after plasmochemy, hyphaes are formed. Later, your fusion of nuclei is produced. This results in a diploid nuclei. The center circles are called as your nuclei. So this results in a diploid nucleate formation. The cells present, on the whole, the cells present inside is called as ascospores. These ascospores are obtained or captured by or present in an ascus, a specialized sac-like structure called as ascus. So during favorable condition, this ascus gets ruptured and it releases your ascospores. And again, they produce the positive and negative strains from which the cycle is repeated. Next come your basidiospore. The basidiospores are the fungi present in it is called as basidiomycetes, commonly known as club fungi. Some examples like your jelly fungi, rusts, shelf fungi, stinking horns, puffballs, toadstools, mushrooms are the best examples, and your bird nest fungi are some of the examples for your basidiomycetes. The basidiomycetes is named after their basidium, a sexual structure that is formed during sexual reproduction, through which your basidiomycetes name has been arrived. This basidium is produced at the tip of your hyphae. Normally, it is a club shaped. Two or more basidiospores are produced in your basidium, and a basidia may held within a fruiting body called as basidiocarp. The basidiocarp is your fruiting body through in which your basidia gets attached. This basidiomycetes can affect humans in many ways. They, as when it comes to your nutrition and distribution, they are saprophytes, decomposed. They present in your decomposed plant debris, especially your cellulose and lignin nutrient-rich areas. They produce a lot. Then many mushrooms are found as food. There are beneficial mushrooms and they are poisonous mushroom. When there is an example we taken for a beneficial mushroom, it is Agaricus campestris, which is an multi-millionaire dollar business in the terms of production and consumption of people nowadays. So it has only one reproduction, which is sexual reproduction, where first your basidiospore formation or germination, where a haploid basidiospore gets uh, produced by your meiosis and it releases or germinates to form a monokaryotic mycelia. There will be two strains, positive and negative. So two monokaryotic mycelia are produced. When fuses by means of your plasmogamy, they result in a dikaryotic mycelium. This dikaryotic mycelium is long-lived in a dormant state as an unfavorable condition is prevailed. Once favorable conditions are coming to existence, they form a button-like structure. From a button-like structure, they develop into a ascocarp. This ascocarp is called as a fruiting body. It consists of, it is the base of the mushroom. So it consists of three main parts. One is your cap, one is your gill, one is your stalk. The cap is called as pilus. Your gill is like the laminae under your cap, or filament, not even a filament, a membrane that is present below your cap. Next, stalk. This it held your cap and gill, from which basidia, the gill which will have your basidia. Okay, the basidia formation of gill, where on the surface of your gills, a specialized club-shaped cells are formed, which is your basidia. Um, First, from which this basidia contains two pair of nuclei, which is a diploid nucleus, which fuses first your plasmogenesis that happens, then your karyogamy occurs. Karyogamy is your fusion of nuclei, where your diploid nucleus is formed from the single cell. A fusion happens, a diploid nucleus is formed. From this meiosis, they undergo meiosis. From a diploid nucleus, they produce four haploid nuclei. Once haploid nuclei are formed, your basidiospores gets formed. So the basidiospore get formed. The basidiospores are small projections, which contain, the small projections are called as sterigma, that withhold or holds your blastospores. So typically every basidium contains four blastospores. Once this occurs at a favorable condition, the sterigma or the spore containing structure gets dispersed into the soil. Again, they produce the basidiospore with a monokaryotic mycelium, then dikaryotic mycelium occurs. This cycle repeats. So this is your life cycle of your basidiomycota or basidiomycetes. Next comes your division of Duromycota. So this is mostly called as fungi imperfectidae. So fungi imperfectidae, and mostly they are present in terrestrial organism, and few being reported they also present in your freshwater or marine. Majority is either your saprophytes and parasites, and parasite they might be on other fungi also. The mostly they affect your human welfare, like example your athletic foot, histoplasmosis, ringworm, etc. So when it comes to your reproduction, the asexual reproduction is by budding, where a bud is produced from your parent cell, and during your favorable condition, the bud gets dispersed into a suitable media or suitable nature, and it results in a new individual formation. When it comes to your sexual reproduction, here it is different. Your sexual reproduction, here it is called as parasexual cycle. It is not normal like your sexual reproduction. It is slightly different. So the first process is your anastomosis. So anastomosis is like They are genetically two different hyphae are present, strain 1 and strain 2, as shown in the image. They first fuse, they come in contact and they fuse, and this process is called as anastomosis. So commonly, first your cytoplasm formation, sorry, fusion, and then your nuclear fusion from both the parent. From there, this results in a specialized structure called as heterokaryon. Heterokaryon. So this fused cell contains two or more genetic different nuclei. This condition is known as heterokaryotic condition. This is the most important step in your this paracycle cycle. Next, in along with this, an unlike nuclei fusion occurs, which is your karyogamy, which is unlikely from a different strain. A diploid nucleus is formed. Okay, this diploid stage is usually short-lived in the fungi. Once this occurs, recombination, the third step occurs. This unlike nuclei fusion and your heterogamy gets fused together. They act as a food cell. Okay, from there a mitotic crossover occurs, or recombination of crossover we might say. So during your mitotic division, there will be genetic recombination occurring within the diploid nuclei. This results in a conidia to form with different colors like yellow or green, as shown in the image. From there, once this recombination or recombination of genetic material occurs, from there haplodization occurs. So haplodization is like it is like your loss of chromosome or reductional stage, we might say, to restore your haploid state. Then from there, a new haploid conidia is formed. Okay, from there, this conidia will germinate into two new, or not even two, new conidial mycelia, which again have a recombinant strain arises. This results, they ensure that there will be genetical variability. From the lastly formed haploid conidia, the sixth step will contain two different strains with different genetical variability. So above all, these are the four divisions, main four divisions we talked about in this presentation of both sexual and asexual reproduction of your fungi. Thank you.